So in this video, we're going to look at DNA structure and how DNA replicates. So first, we need to know that DNA is made out of repeating nucleotides. So this structure here is called a nucleotide. And it has three parts. It has a negatively charged phosphate group, a sugar called deoxyribose, and a nitrogen base. So when we say that DNA or nucleic acids are made of CHO, N, and P, the sugar is CHO, the nitrogen base is CHON, and then that phosphate is found up here in the phosphate group. And this nitrogen base has A, T, C, or G. We have purines and pyrimidines. Now, as a human, our genome is made out of 3.2 billion of these nitrogen bases. And these nitrogen bases, as monomers, hook together to form, or pair together to form a polymer. So DNA is a polymer, a nucleic acid, made out of these nucleotides. So when you see something like A pairs with T, it's really a nitrogen base A pairing with another nitrogen base T. And when we talk about base pairing, A will always pair with T with 2 hydrogen bonds, and C and G pair together with 3 hydrogen bonds. So there's hydrogen bonds that are between the bases. And so later on, when we see how DNA replicates, it's these hydrogen bonds that are going to be broken. Now the cool thing about DNA is that if we know one strand of DNA, we can find the complementary strand because of base pairing rules. So here we have that right there alone is how you do basically replication. What we're going to see later on down here is when you separate these two strands, you will use one side to build the other side. So DNA, kind of cool, is that it runs anti-parallel. If you want more information on this, you need to watch my other YouTube video that explains DNA replication in detail. So when we say anti-parallel, that means that you have a 5' end and a 3' end at one side, and then 3 and 5. So they're run in opposite directions. So this is important for replication. So in DNA replication, if we start here with the strand of DNA, you can see that the 5' and 3' ends are opposite of each other. You can also see that they're held together by hydrogen bonds. Now the bond between a G and a T, like in the backbone, so like here, the bonds would be phosphodiester bonds, covalent bonds. So in order to replicate DNA, we need to separate these two strands. Our DNA is made basically of semi-conservative replication. At the end of replication, we'll have 2 exact copies of DNA. And you can see here in black, that was our original strand right here. And then we use that code in one strand as a guide to build the complementary strand. Because of base pairing rules, if we know one side, we can figure out the other. So when we look at these molecules, you'll see it's semi-conservative. Here's one half of the original, and then this light blue represents the new strand that was built. So there's a series of enzymes involved in DNA replication. First, we have an enzyme called Topoisomerase that kind of loosens and takes the pressure off of the double helix, making it easier for the enzyme helicase to break the bonds between base pairs. So helicase is an enzyme that basically unzips or breaks the hydrogen bonds. It'll be moving in this direction. That is the direction of the growing replication fork. So here, as helicase has come and it's unzipped and broken these bonds, what we find is an open strands of DNA. Well, we need to build the new complementary strands, keeping in mind that the new strand is going to be anti-parallel. So up here where we saw DNA running in opposite directions, when you build the new strand, it's also going to be anti-parallel. So if this is the 5' end of the existing strand, then right here is the 3' end of the new strand we're going to build. Here's the 3' end of the existing and the 5' end of the new strand. So when DNA replicates, it uses the main DNA builder, I guess, is called DNA polymerase 3. DNA polymerase 3 is the enzyme that's going to add complementary nucleotides. So it's represented by this green highlight. Pretend that's an enzyme that is adding this T, adding this T to this A through hydrogen bonds. So you'd have two hydrogen bonds right here. Now, the thing about DNA polymerase 3 is it can only build from 5' to 3'. It can only attach on to this 3' end. Again, if you need a refresher on this, I'd watch my other video about DNA structure. It'll explain this in more detail. So here, in order to start, the other thing about DNA polymerase 3 is it needs an existing 3' end. So with these nucleotides, the 3' end is right here. It needs an existing one to attach to build the new phosphodiester bond. So to attach and build a new nucleotide, it needs an existing end. So therefore, before replication can begin in DNA, there needs to be a primer. So the primer, though, is built out of RNA. So here you have an RNA primer, and the enzyme primase builds that RNA primer, and now there's an existing 3' end for RNA polymerase to begin and attach. So, because, did I say RNA? I mean DNA polymerase. So here how you see that DNA polymerase builds from 5 to 3. In this new strand, this is the 5' end. Therefore, DNA polymerase can build from 5 towards the 3. So if here is the 3' end, that means that this strand is going to, once it gets started off of the primer, will be built continuously as the helicase moves. So like here, once it starts with that one primer, it can just follow helicase. As helicase unzips, then more additional nucleotides can be added. Now, on the other side, the new strand has a 3 right here. Well, DNA polymerase 3 can only build, and 1, can only build from 5' to 3'. That means it can only attach on to this side. That's opposite of the replication fork moving this way. You'll notice that this also requires a primer. The primer will be put here, and then DNA polymerase 3 can build from the 5' to 3' ends. So when we look at how the DNA is replicated, you have this leading strand where it started with one primer and it follows the helicase. Now in the lagging strand, it's built in the opposite direction of the replication fork. So as helicase moves in this direction and it unzips, it exposes a new section that will be open. So like the next fragment would have an RNA primer here, and then it would be built from 5 to 3. It would stop wherever it hits that previous fragment. So this strand in light blue is built in the opposite direction of the replication fork, still being built though from the 5' end to the 3' end, 5' end to 3' end. So it's built in Okazaki fragments, and each of these fragments requires a primer. Now, once the fragments have been built, we need to replace those RNA nucleotides. We need to take out those RNA nucleotides and replace them with DNA nucleotides. And that is the job of the enzyme DNA polymerase 1. Now, DNA polymerase 1 will also scroll through, basically, and proofread and check for errors and mistakes, trying to prevent mutations. But DNA polymerase 1 will remove that RNA primer, replacing it with DNA nucleotides. However, it too builds from 5' to 3' and cannot attach the 5' end. So right here, this primer, when it's removed, actually will not be replaced because it's the 5' end, and therefore your chromosome gets a little bit shorter. Over on this end, though, each of these primers will be removed and replaced with DNA nucleotides by DNA polymerase 1. Then we have an additional enzyme called ligase, and ligase will come and seal these fragments and build the phosphodiester bonds between them. And then that will have a solid strand there. So over time, you end up with 2 strands that are part of the original, which is the black ink. And then here you have the light blue represents the new strands that were built by 5 or so different enzymes following base pairing rules. And we call this semi-conservative replication.